Today we are looking at the history of microbiology. Uh, we want to look at, we want to understand where humankind is coming from and where is it going in terms of our understanding of microbes. The things that we know today um, might seem very obvious, but these are things that many years ago um, mankind never knew. Okay, these days you go to the hospital, very fast you'll be told you have an infection. And of course, we know we can use different antimicrobial agents to treat these infections, whether it is a fungal infection, bacterial infection, viral infection, and you're treated and you are good. Um, so that seems to be very easy, right? But in ancient times, the understanding of what even causes infections, that understanding wasn't there. Um, and then the understanding that we can treat these infections with these um, antimicrobial agents was not also there. So people used to die from simple infections, things that we can call simple infections now. We actually call them simple infections now or mild infections now because we know how to treat them and we know their pathogenesis. Um, However, the challenge that we are facing in this generation is the issue to do with antimicrobial resistance to drugs. So every generation has its own challenges. So that is the challenge that we are facing. In ancient times, the challenge was what are the things affecting and killing a lot of people? We don't know them. How do we treat them? Now we know what can be used to treat what condition, but we are misusing the drugs, and as a result, we are having antimicrobial resistance. So, or even more specifically, you can say antibiotic resistance. But you also know that also viruses are developing resistance. Also, they have we have antiviral resistance. Okay, so we want to understand the personalities involved and what is their contribution. And some of these stories are actually very interesting because there is one or two lessons that you can actually pick from these personalities and their discovery. So that's what we are going to do in this presentation. So yesterday, I don't know whether you paid keen attention to what we termed as the evolutionary tree. We looked at the three domains yesterday. So those three domains, there was a picture on that slide that shows the different domains. Picture was illustrating what we call the evolutionary tree or the ancestral tree, whatever you can call it. You realize that the level at which these different domains are branching off and the different categories or groups of organisms. That diagram is not by mistake that it was like that. It was to illustrate that our scientific understanding is that not all organisms appeared on Earth at the same time. Okay, not all organisms on Earth appeared at the same time. So you have the emergence of different species over time. Okay, through a process that we call speciation, if you remember that biology. So here we are saying that bacterial ancestors were the first living cells to appear on Earth. So it means they are the oldest on Earth. And for the most of human history, people actually knew little about uh, these diseases that were actually affecting people, uh, how they are caused, how they are transmitted, and how to treat them. Okay, so microbiology actually is a field that helps us to go into that history of transitioning from when people didn't know these microbial agents to the current things, microbial technologies that we are applying, issues to do with uh, molecular biology, genetic engineering, development of vaccines, and all those are modern microbial techniques. But how did we arrive at that point where we are having all this advancement? So microbiology is what can help us understand that, especially the history. So the first personality that you're going to talk about is Robert Hooke. So Robert Hooke used the microscope to look at the back of a tree, and they saw some small squares, just like what you see there. So he concluded that living organisms actually have small units. He called them small boxes. Um, and of course, as we know them, these are cells. So that actually gave rise to what we call the cell theory. So the cell theory is that all living organisms are composed of cells. That is the hallmark of what we call the cell theory. Okay? You cannot have a living organism that is not made up of cells. Okay, so that was the start of starting to look at things that we cannot see with our naked eyes, but when we use a microscope, we can see something. So of course, that activated curiosity among different people to start using these microscopes such that they can be able to see things clearly, and then set the stage for further developments. Remember that though people didn't know what causes infections or diseases, people speculated. There was speculation. So they had different theories to do with what we call the miasma theory, theories to do with the fact that, you see, people get diseases because of body odor, it is punishment. All the theories existed. Now, the first person to think about the fact that microbes actually contribute to disease causation is this person here, Athanasius Kircher. Okay, he's the person who linked microbes to disease development. That is what that was his contribution. Okay, so his contribution meant that people now wanted to generate evidence to prove whether he's right or he's actually wrong. So that lays a very good foundation for further scientific research in medicine and biology. Okay, so how did he arrive at that conclusion that perhaps microbes contribute to disease development? What he realized is that using a microscope, he observed blood, and in that blood of victims of plague, he saw tiny worms this blood. So he linked these tiny worms to the disease. So he said, or he concluded that these tiny worms are linked to the disease. Okay, and that laid the fertile ground for the development of what we call the germ theory. Okay, where we talk about microbial agents actually contributing to disease development. Now, time came when discoveries in microbiology were actually um, becoming very rapid. One discovery over the other, one discovery over the other. So, um, starting with, you can see that in our presentations we have started with the microscope playing an important role almost for each person who we are actually talking about here. So one of those personalities that actually is very fundamental in microbiology is Anton van Leeuwenhoek. He was a Dutch national. Okay, so what did he do? Using again the microscope. Now, with the microscope, the people who started using microscope from Robert Hooke, the microscope was not advanced, so it could not see a lot of a lot of detail. Okay, so what he did actually was to develop a lens that could magnify specimen to up to 300 magnification. So that means that he was able to see things in more detail compared to other people. So that meant that he could provide detailed description of organisms that he would see. So what did he see or discover? He discovered things that we now know as bacteria, yeast, and protozoa. For now, we call the things as microbes or microorganisms, but of course, at his time, those words were not actually there. So words like animalcule could be used to describe what he was saying. And the description that he used, and you can see this is a statement. I saw with great wonder that there were very many, very little living animalcules. Very prettily are moving. I was explaining what he was writing. This phrase here. If you write like this, currently, it may not be what we term as scientific writing. Okay, so that description that they had, that writing that they had, was more of descriptive, trying to bring the picture of what they were actually observing. Okay, so he observed a number of specimens, including tooth scrapings, pond water, and blood, and documenting detailed morphological shapes that transformed our lives over the years. Okay, so these people were actually very descriptive, and through being descriptive, that is where other people can pick up. And this is also to tell you that science is you build on other people's work. Every scientific writing or discovery is based on other people's work, and therefore, if you provide very good descriptions, it means other people can easily pick up and continue with what you have been doing. One of the big debates was the debate about the origin of life. If you go back to evolution in biology, there's that topic called evolution. Okay. Um, so under evolution, one of the biggest questions that we have there is what is the origin of life? So there are different theories about origin of life, and uh, the topic, I mean, the theories that we have in here, we can call them theories or hypotheses. We have one that is termed as the spontaneous generation theory of origin of life, and then biogenesis. Now, the first one is, of course, I hope, let me try to clarify what the difference between a theory and a hypothesis. So with a hypothesis, you make a statement. You just make a statement. That statement can be true or false. Whereas a theory comes from a statement. So how does it come from a statement? You start with that statement, of course, is the hypothetical statement. Or you write your hypothesis, and that hypothesis is what we term as the hypothesis statement. So that statement, you don't know whether it is true or false. Now you carry out experiments, and through these experiments, if you have consistent findings, you can get a theory. Okay, so that's why these ones we're actually calling them hypotheses, though in other sources they can be categorized as the theories about origin of life. So spontaneous generation, for it believed that life came, or life arises spontaneously from non-living matter. That if you have bread and you leave it there, that bread is non-living. You come after 2 or 3 days, you'll find some growth on it, and most likely it will be fungi. So for them, that is the people who are proposing this theory or hypothesis, that's what they believe, that every living organism that we actually have came from something that was non-living. Just like when you leave food that is uncovered, flies come and they lay eggs, and then after a few days, you see maggots there. So that food is non-living, and then you see maggots appearing there. So for them, they say, okay, that is how microbes actually arise. And then the biogenesis hypothesis, for it stresses one fact, that all living cells actually have to arise from living cells. So you can see that it is in opposition to the spontaneous generation hypothesis. And we believe this today, after today, that of course every living organism comes from cells. Every living organism, any cell that you have, it must be coming from a pre-existing cell. Okay. Now, of course, different experiments have been done to either prove or disprove some of these hypotheses. And one person, Francesco, performed what is commonly known as the Rudi experiment. So with the Rudi experiment, what he did is that he got jugs and filled them with meat, and then left them. You have one jug that is open. It is not closed, and then after some time, the maggots are there. Okay, and then another jug, the meat is there and it is covered. It is well sealed. You can see this, and there are no maggots after some days. And then people said, okay, the explanation here is that people believe that organisms actually came from air. Okay, they came from air. And now if you have sealed it, if you have covered it, you don't have air there, and therefore you don't have sufficient what? Oxygen for these microbes actually to grow here, to appear. And then he said, okay, fine, let me get a seal. Let me seal off, but allow air to enter. So he got a gauze. So this gauze allows air, and that means that the air could reach the meat, but again, you had no what? No maggots, yet the air was actually present. So that means this experiment proves that what we disproved what people were basically saying, that if you have air, you will have living organisms, I mean microbes. That's what it proved. Now, other experiments also. Needham did an experiment. For him, he heated what we call broth. Broth is a nutrient media, which is liquid. When we talk about nutrient media, we are referring to the microbes need nutrients for them to grow, divide in number, and increase in number. So they need nutrient. So these nutrients we can supply, or we can control the nutrients in the laboratory, and then make sure that some of these microbes can grow. When you do that, that is what we call culturing. So that means that you need the nutrient media. We have different nutrient media that we can use in culturing or growing these microbes. So one of those is what we call broth, which I'm saying is a liquid media. Okay, so John heated it and poured it in a covered flask, in covered flasks. So after some time, again, microbes appeared. So for him, he believed again, like what other people are saying, that microbes developed spontaneously, because by heating these microbes, he thought he has killed all these microbes, and therefore, after some time, you cannot see these microbes in these flasks, but they appeared. So his conclusion or claim was that the microbes actually appeared spontaneously. And then another person also did another experiment. For him, this experiment is actually related to what John did. That is Spallanzani. For his experiment, which you can see that it is 20 years later. You can see this one is 1745, and then this one is 1765. For him, he suggested that the microbes is actually providing more of an explanation to this experiment. Suggested that the microbes from the air entered after boiling, and that if you seal the flasks after boiling, you will actually have no microbes. So you will have a sterile, the culture will not have any microbe. So again, Spallanzani was criticized. Why? For John Needham, for him, he claimed that, of course, for him, he boiled, and then after boiling, again, you have microbes there. For him, he claimed that there was a vital force that was destroyed by the boiling or by the heat, and kept out by the seals, if covered. That was his explanation. And then critics argued that there was no sufficient oxygen in the sealed flask to support life force. So that's why you cannot see microbes. if you seal off the broth flasks. Of course, these debates continued for another century, and again, the theory about the fact that living cells arise only from pre-existing living cells continue to predominate. Okay, and Rodroff was a proponent of this, but of course he lacked the experimental evidence to kill the debate off. Remember, we still had a number of debates on where these microbes actually arise. And now we come to Louis Pasteur. Now this you can term this era where when we start talking about Louis Pasteur, this is where we can describe this era as the golden era of microbiology. The golden era of microbiology. Things at this point actually started becoming very, 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 very clear. And remember, we hinted on the germ theory, where we are linking microbes to development of diseases. But of course, at that time, the experimental evidence was not actually very sufficient. And now people like when people like Louis Pasteur actually came in, the germ theory took center stage because he performed the experiments that actually provided evidence that you have organisms that are causing diseases. And that is basically the basis of the germ theory. So the emergence of the germ theory killed the hypothesis about spontaneous generation. So through Louis Pasteur experiments, the spontaneous generation hypothesis was actually disproved. So what are the things that he actually did? His experiments were not actually very different. You can see that these experiments they have resemblance to the experiments that other people that we have described actually carried out. Again, you can say that we are talking about here we're talking about effusions. Effusions, these ones, nutrients, infusions that contain nutrients that can allow microbes to actually grow. Again, you can see that the experiment is actually designed in flasks, and we have seen flasks repeatedly. The unique thing with his experiment was that the tubings that connect to the flasks were actually not straight. They are S-shaped. Now, that is actually what makes his experiments actually different here. So when you boil when he boiled the infusion, this one boiling, we know as of now that boiling kills microbes, many of them. So the infusion was boiled for a long time to kill all the microbes that are actually present. But since these flasks are not sealed, it means that air actually enters these flasks. But since they are actually in S-shaped, these tubes are in S-shaped, it means that the microbes actually settle somewhere. And in this case, they settle here. So they don't reach this place. So this actually remains clear. Okay? And when he tilts, when this flask is tilted, the microbes which had settled here actually can move and get entrapped here. And this definitely gets, you can see changes because you have microbes in there. Okay, so the microbes could quickly multiply in this infusion. Okay, So what that experiment proves is that in the air you have microbes, but the air itself does not create the microbes. So within the air you have microbes, but the air itself does not create the microbes. So this disproves the spontaneous generation hypothesis. So microbes do contaminate liquids or solids, and they can come from air. So within the air, you have microbes. If you heat the infusion, you kill the microbes. We also know that even now. And we use the basis of our understanding of the fact that we have air, we have the air that we have around us contains microbes. That's why we put on some masks if we are suspicious of dangerous microbes. But also, if we want to decontaminate some instruments, we do heat them. And these are the basis, it actually forms the basis of asepsis or aseptic techniques, where we talk about autoclaving, steam sterilization, and things like that. So there's this statement here that the original flasks that actually Pasteur used are still available in the what is termed as the Pasteur Institute. The lesson that we have here is that can we preserve the most important things? Okay, the most important things, the contributions, the groundbreaking contributions that are made by people, these things should be preserved. Okay, so on the basis of Louis Pasteur's work, we can do a number of things. We can carry out fermentation, and fermentation is very important, whether at home level, homestead level, or even at industrial level. The production of alcohol is through fermentation, and it is through Louis Pasteur's work that the role of microbes in fermentation was actually discovered. The way we preserve liquids, especially milk, pasteurization is one of the ways, which is basically by heating these liquids, we kill the microbes, and we can preserve or make sure that the milk does not have microbes that can cause diseases. And of course, his work also emphasized the germ theory, which linked microbes to diseases that happen. So issues to do with the autoclaving that you have in your hospitals, these ones are actually as a result of Louis Pasteur's work. So the germ theory is what now we actually know that diseases are not happening because God is punishing people. Diseases are not happening because of foul odors, or diseases are not happening because of poisonous vapors. Diseases are happening because some diseases are happening because you have microbes that damage tissues. And in here, the Agostino Bassi linked silkworm disease to, now we're talking about real organisms now, fungus. Louis Pasteur also linked silkworm disease to a protozoan. But the whole thing is that, or the whole germ theory thing is that you have microorganisms contributing to changes in the living organisms and causing diseases. Now, this one looks very simple. A simple task as hand washing, and I think we have what we call the Global Hand Washing Day. Hand washing, as simple as it was, people didn't know that hand washing can actually reduce mortality. So Ignace Semmelweis is the one who discovered this. Now, how did he discover? He was in a teaching hospital. So in a teaching hospital, you have different health professionals being trained: nurses, midwives, doctors, even physicians who are basically upgrading. So we observed one thing, that in the maternity ward, where the doctors actually work, there were increased number of mortalities. And then where the midwives actually work, the mortalities are not very high. So he asked himself a question: why? What he discovered was that the physicians actually, they were touching patients and they were not washing their hands. And the midwives were not doing this. Okay? So different people doing different things. So he said, please, the physicians, wash your hands, because the midwives are washing their hands and their patients are not dying. The numbers speak for themselves. These people did not believe what he was saying until actually his death, that people realized that what actually Ignace was talking about was actually true. Okay? So he advocated for hand disinfection, but most of his colleagues refused the idea of hand washing, and patients continued to die. He tried to do the advocacy, but he was not very successful. So we move to Joseph Lister. For him, he introduced the use of antiseptic to treat wounds and sterilize surgical instruments. So again, we know when you're preparing a patient for surgical interventions, we need to do drape, proper draping, proper gloving, proper aseptic techniques, and then the cleaning of the area using iodine and all these antiseptics that we have. This was not something that was being practiced, and we can attribute that to that revolution to Joseph Lister. Okay, so he introduced the use of phenol, which is basically carbolic acid, as an antiseptic to treat wounds and sterilize instruments, and this reduced the number of post-operative infections that are happening as a result of making sure that things are done aseptically. Vaccines come into the picture. Now, like I said earlier on, things don't emerge from nowhere. Things, people do research in certain areas, and other people come and pick from what other people have contributed. Now, Louis Pasteur developed a vaccine against anthrax. We know that anthrax affects humans and also livestock. It's actually a dangerous condition. So what he did, he used what we term as attenuated organism. Attenuated organism is an organism that has been weakened, and then you introduce it into the person you want to immunize, and then it helps to boost the person's immunity such that they can fight off the condition. And that is what we continue to use. Now, Robert Koch, for those of you who have done epidemiology, you must have seen, you must have looked at Robert Koch. If you didn't, you need to go back to those slides and check. Do you have Robert Koch in the history of epidemiology? So what did Robert Koch do? So you can see that from Louis Pasteur, we are talking about people who have discovered organisms, particular organisms. We said that Louis Pasteur discovered a number of them. We talked about bacteria, we talked about protozoa, and the last slide we have emphasized that he discovered the bacteria that causes anthrax. Now here, moving ahead, we shall see personalities that are now discovering different organisms. So from Louis Pasteur, we now move to Robert Koch, and we are saying that for him, he discovered the number of these organisms. Tuberculosis bacillus is one of those organisms. He isolated it. Bacillus anthracis, the one that causes anthrax, he isolated it. And then the one that causes cholera, Vibrio chloride, he also isolated it. Okay. So here, what is notable here is that you get samples or specimens that contain these microbes and be able to separate that microbe where you have grown it or cultured it and be able to visualize it clearly. That is what we are basically meaning by isolating. You culture and then isolate. Now, one of the key things about Robert Koch is he was a German. So what he came up with, what we term as the Koch postulates. These are four fundamental criteria that he laid down, that if you follow these things, you are very sure that a particular microbial agent is the one responsible for causing a particular disease. Okay, it's like saying that if you follow these four things, you will know who has stolen your phone. If your phone has been stolen in you're a group of people, and then among those people there is someone definitely who has stolen your phone. If you follow these four things, if they are there, you will be able to identify who has stolen your phone. It's the same thing that Robert Koch is actually putting across here, that if you follow these four things, the four postulates, you'll be able to link a causative agent to a particular disease. So these four things are: one, that microorganism that you are suspecting to cause a particular disease, it should be there in a person who is having that disease. That is postulate number one. Okay. If you say a patient is having malaria, if you get that patient's blood, you should be able to get the malaria parasites there. If you don't see the malaria parasites there, that is not malaria. Plus, number two, that the pathogen can be isolated from the diseased host and grown in pure culture. If you suspect that a person is having a bacterial infection, whether that bacterial infection is very severe and that you can get that patient's blood and still get the bacteria there. If you get this patient's blood or specimen, it can be sputum even, sputum. If you get this sputum, you go and analyze it, you should be able to get the bacteria. And when you get that bacteria or that pathogen, if you get this sputum, go and get this pathogen, and then be able to grow that pathogen such that it multiplies the number such that you can see it. That is point number 2. Point number 3. Now, you have obtained the pathogen from the patient's specimen or from the host's specimen, and you have grown it, and it has grown. Now, the pathogen from the pure culture must cause the disease when it is introduced in a person who is healthy or in a host that is healthy. So you get what it basically means, that you have gotten this sputum. You have isolated in this case, let us use bacterium as the pathogen, and you have grown it through different culture. When you get the bacteria that you have grown and put it in a person who is healthy, that person should get the disease that pathogen causes. That's point number 3. The last one, the pathogen must be re-isolated from the new host and shown to be the same as the originally inoculated pathogen. Inoculating is introducing the pathogen in a particular, it can be media or it can be a host. That is inoculation. So if you get this pathogen from this new host that you have infected, and this new host that you have infected actually has developed the disease that is caused by this pathogen. Now, when you get another specimen from that person, that's new host, and you go again and analyze and look at it, it should be the same pathogen. It should not be another pathogen. So that is Koch's postulates. And when you realize that these are the same things that we actually use in our practice, that if you go to the hospital, they test you, they find that this one is not there, please, even if you ask for medication, no trustworthy physician will give you the medication for a condition that they have tested and they have seen that you don't have it. You go for an HIV test, they say you don't have HIV, they cannot start you on ARVs. Okay, because we can easily link HIV infection to the microbe, the discovery of the HIV virus, the HIV in your blood is an indication that you have the infection. So you can start the treatment. If you don't see these viruses, whether the antibodies, we cannot start you on this medication. So as a result of these postulates, we can be able to know that, we can apply that, and we can link particular microbes to specific diseases. We know that hepatitis B is caused by this one is caused by this was caused by that. We can also differentiate between organisms that can cause diseases and those that cannot cause diseases. The ones that cause diseases are the ones we call pathogenic. Those that don't cause diseases are called non-pathogenic species. It also allows us to do classification, and we looked at this classification yesterday. And then we can also, based on this knowledge, to be able to carry out experiments to determine whether you have resistance or no resistance, especially if you perform these experiments in laboratory animals like rats. Again, the story of vaccines is surfacing. One of the key, if they ask you at any point in time, what are the successes in public health? The successes of public health include the discovery of microbes that they are causing diseases. Another one is that the introduction of antimicrobial agents like antivirals, bacteria, and things like that. Another success of public health is the discovery of vaccines. So the discovery of vaccines is very, very important. So before the discovery of vaccines, people were dying with no options. Okay. But of course, people were not folding their hands because there are no vaccines. Different things were being practiced. Okay. So what are those things? The Chinese actually used the powders from pustules to be able to prevent other people from getting some of these conditions, like smallpox. And you see that it's the basis of what we actually do now. That, of course, this was crude with what you can term as scientific basis, but what we know as vaccines now, the ideas arose from things like this. So a pustule is like a small swelling. So the extracts from these pustules are what are given to people, and then they somehow survive. And then people are like, okay, this actually works. Okay, so this is what you call variolation. You get specimen or extracts from one person and introduce it to another person, get these extracts, introduce them in another person with the belief that they will not be severely affected by a condition. Okay, that is variolation. Now, you cannot talk about the history of vaccines without Edward Jenner. If you don't mention this person, you have not given a right description of the history of vaccines. So this person also appears in the description of epidemiology, the history of epidemiology. So Edward Jenner, what did he do? So he observed that people who got cowpox were not affected by smallpox. Such is what he saw. So cowpox is a mild condition that was affecting people who were milking cows. So these people were not affected by smallpox. So he wondered, could it be that this cowpox protects them from smallpox? So what he did, which is similar to what we have just seen with the Chinese, was that for him, he scrapped the cowpox blisters and got the extracts and inoculated. You can see that we are using the same word again. Inoculated these extracts into an 8-year-old boy. And actually, this was his boy. Okay. And the boy was protected from smallpox. Okay. And the word vaccine actually has origin has a Latin origin. Vacca, which means cow, is related to the cowpox experiment that Edward Jenner actually carried out. For him, he saw that people who are having cowpox were not severely affected by smallpox. So these blisters, get scrapings from these blisters, and if you introduce this to people, they get well. So why not? So you can see, again, what you can see with this is the ethics. If we ask you the ethics, the ethics related to this experiment. The ethics that we know that for you to perform such an experiment, you need to get approval. Previously, the things were not there. Okay, the things were not there. And he did it on his own child. How ethical is that? But it worked. Okay. Again, about vaccines, Pasteur discovered why vaccines work while studying fall cholera. So what did he do? What are the things that he actually saw? The term virulence actually comes in. And that person with a question, we move. The word virulence is the word that we shall encounter when we are looking at immunology. Immunology is one of the topics that we shall look at in this module. So, virulence is related to the capacity of organisms to cause infections. Now, a virulence, it means the organism is not going to cause infection. Okay, so what Pasteur discovered was that if you have cultures that have overstayed for long, the microbes there will not be able to cause diseases. So they are virulent. Remember, virulence is the ability of the organism to cause a disease. When you grow these organisms that can cause a particular disease and they stay for long, they may not cause disease. So that's what Louis Pasteur discovered. And then another concept that is also very important, and it is also what we apply now, is that weakened organisms can actually protect against virulent counterparts. And if you use the word virulent, we know what you're talking about, the ability of the organism to cause a disease. So more virulent organisms can have a higher ability to cause disease. Now, if you have weakened strains, that the variety that are weak, they prevent the more virulent ones from causing a disease. That is the basis of vaccines. That's how vaccines work. And that is why vaccines are made from weakened or killed microbes, or even from genetic engineering. So when we ask you a question, where do we get vaccines? You should be able to tell us that these vaccines can be made from weakened organisms. These vaccines can be made from killed organisms, or these vaccines can actually be made through what you call genetic engineering. John Tidal. So what did he do? Again, it is related to the discoveries that we had from the start, that we can kill microbes by heating them. So for him, he discovered that, okay, you can kill, but you cannot kill all. Why can't you kill all? Because some of them, as we know now, some of them can produce spores. And some of these spores are actually resistant to heat. He also observed the antimicrobial properties of mold penicillin. Antimicrobial properties of the mold penicillin. Decades even before Fleming, in our, I think even primary, they ask us who discovered penicillin, and you talk about Fleming. Okay, so for him, he discovered this way before Fleming actually discovered penicillin. It appears there is a person who has raised up their hand. The reason as to why I'm going very slow with this presentation is that it will form the basis for your understanding of the other presentations. So later on, I'll try to increase the speed at which I go through these presentations. So Christian Graham, what did he discover? The things that we talk about in our clinical area, I mean gram positive or gram negative bacteria, it's about this gram stain that is carried out in the laboratory. Even the most basic laboratories, I think, can perform this gram stain. So what is gram staining? It's about applying stains, or basically dyes, and certain bacteria will retain the color, and then certain bacteria will get decolorized. And that is as a result of the structure of the cell wall that these bacteria have. Peptoglycan that we talked about yesterday, it is in the gram-negative bacteria, it is very thin, and in gram-positive bacteria, it is actually very thick. And as a result, the staining is actually different for the bacteria that have this thick peptoglycan layer, and then for the the bacteria that just have a thin peptoglycan layer. Okay, so that is the gram stain. We shall look at it and know the implications. The implications there, it is performed in clinical area to know what am I treating? Is it a gram-positive bacteria, or are we treating an infection that is caused by gram-positive bacteria, or is it an infection caused by gram-negative bacteria? Because the basis for which we have what we term as narrow spectrum antibiotics or broad spectrum antibiotics, it's from this. If you talk about broad spectrum antibiotics, they can deal with the gram-positive bacteria and gram-negative bacteria. And then those that are very specific, they cannot kill these two categories of bacteria. So that's why staining is actually very important. The viruses that we talk about today, we are basing on the work that was done by Martinus Bengerick. He's the one who discovered virus, and he's the one that gave that name because they were smaller than the bacteria. He discovered it through tobacco mosaic disease. That's when he isolated. So he's considered one of the primary founders of virology. Another person that we can talk about here is Paul, and his discovery is about a drug that could hunt down and destroy a pathogen without harming the host. And he used arsenic derivatives that were actually effective against syphilis. And it's the first true synthetic drug to be made. So this was considered to be a magic bullet. You can see the years, 1910. So we are moving to a phase of talking about drugs now. Remember, if you have been following our slides critically, we moved from a phase of what is causing diseases, and then we moved to a phase of could it be microbes? If they are microbes, where do they come from? And then we moved to the discovery of the actual microbes now. And then now we are talking about discovery of antimicrobial agents, things that can kill these microbes. So this is the phase where we are. And Fleming comes in automatically. And we think we had talked about him. So you can see the first drugs that came up, quinine, derivatives of dyes, and sulfonamides, which are antibiotics. You can see 1930s now, talking about drugs. Fleming's talking about penicillin. coming from fungi or mold. Okay, and is considered the father of modern chemotherapy. When we talk about chemotherapy in microbiology, you're talking about using agents to kill microbes. We can chemotherapy or antimicrobial agents. So he's Alexander Fleming is considered to be the father of modern chemotherapy. He marked the birth of modern chemotherapy. So, how was the journey?